Hey guys, welcome to my channel Nature Girl Priya. Today I am going to explain you about how to grow new plant with the quality you want to see in the plant using different plant cells form different plants. We are going to talk about some interesting facts about it, like frog nerve cells were first cultured successfully, and more than 32,000 papers rely on cell lines with no known original stock. So to know about all this, watch till the end. And with that said, let's roll the intro. You must be knowing who is the father of plant tissue culture. It's Haviland who conceived the concept of cell culture in 1902. Now let me explain what tissue culture is. Tissue culture refers to a method in which fragments of a tissue, both of plant or animal tissue, are introduced into a new, artificial environment, where they continue to function or grow. The media used for the growth of the culture is broth and agar. A tissue culture typically takes 10 to 14 weeks, beginning with clipping very small leaf, stem and root samples to obtain cells and then letting them establish themselves in a nutrient mix, where they eventually multiply. Those cells grow into leaves and stems without roots. Tarankan. While the term tissue culture may be used for both plant and animal tissues, plant tissue culture is the more specific term used for the culture of plant tissues in tissue culture. This technique is also known as micropropagation. It has proved beneficial for the production of disease-free plants and increase plant yield in developing countries. It only requires a sterile workplace, greenhouse, trained manpower, and a nursery. Oil palm, banana, plantain, eggplant, pineapple, rubber tree, tomato, sweet potato have been produced by tissue culture in the developing countries. Tissue culture is also known as cell culture, plant culture, organ culture, plant tissue culture or sample. There are two main types of cultures. Primary culture, it consists of normal tissues excised from the living organisms by biopsy. They model the natural function of the tissue. These cultures are generally motile. Cultures of established cell lines, these are either derived from the tumor biopsies, or from the primary cells that had undergone mutation and continued to replicate. There are different types of tissue culture techniques. They are seed culture. In this culture, the explants are obtained from an in vitro derived plant and introduced into a laboratory where they proliferate. The explant should be sterilized to prevent it from tissue damage. Embryo culture. This type of process involves in vitro development of an embryo. An embryo is isolated from a living organism. It can be mature or immature embryo. The ovule, seed or fruit is already sterilized, therefore, it does not need to be sterilized again. Callus culture. A callus is an unorganized, dividing mass of cells. When the explants are cultured in a proper medium, the callus is obtained. The growth of callus is followed by the organ differentiation. The culture is grown on a gel-like medium composed of agar and specific nutrients required for the growth of the cells. Organ culture. In this, any organ of the plant such as shoot, leaf, can be used as an explant. A number of methods can be used for the organ culture, such as plasma clot method, raft method, grid method, and agar gel method. This method is used to preserve the structure and functions of an organism. Protoplast culture. It is a cell without a cell wall. A protoplast can be cultured using the hanging drop method, or microculture chambers. In protoplast culture, a number of phases can be observed: development of cell wall, cell division, regeneration of a whole plant. Other types: pollen culture, anther culture, single cell culture, suspension culture, somatic embryogenesis. Phases of tissue culture. Number 1. Initiation phase. At this stage, the tissue is initiated into the culture. The tissue of interest is obtained, introduced and sterilized to prevent the process from any contamination. Number 2. Multiplication phase. In this stage, the sterilized explant is introduced into the medium composed of growth regulators and appropriate nutrients. They are responsible for the multiplication of cells. This undifferentiated mass of cells is known as callus. Number 3. Root formation. The roots start forming. Plant growth hormones are added to initiate the root formation. Consequently, we obtain complete plantlets. Number 4. Shoot formation. The plant growth hormones for the formation of shoot are added and the growth is observed for a week. Number 5. Acclimatization. When the plant starts developing, it is transferred to the greenhouse to develop under controlled environmental conditions. It is finally transferred to the nurseries to grow under natural environmental conditions. The process of plant tissue culture. This process involves the use of small pieces of a given plant tissue. The tissue obtained from a plant to be cultured is called an explant. Explants can be taken from many different parts of a plant, including portions of shoots, leaves, stems, flowers, roots, single undifferentiated cells, and from many types of mature cells provided they still contain living cytoplasm and nuclei and are able to differentiate and resume cell division. Once the tissue is obtained, it is then cultured in the appropriate medium under sterile conditions so as to prevent various types of microorganisms from affecting the process. For doing tissue culture, there are some general procedure which involves medium preparation, plant preparation and transfer of plant material to a tissue culture medium. Let's discuss about this briefly. So first, medium preparation. Number 1. The appropriate mixture is mixed with distilled water and stirred while adding the appropriate amount of sugar and sugar mixture. Here, sodium hydroxide or hydrochloric acid is used to adjust the Fe contents used here will depend on the plant to be cultured and the number of tissues to be cultured. Number 2. Agar is added to the mixture, heat and stir to dissolve. Number 3. After cooling, the warm medium is poured into polycarbonate tubes to a depth of about 4 cm. Number 4. With lids sitting on the tubes are placed in a pressure cooker and sterilized for 20 minutes. Next comes plant preparation. Number 1. Cut the plant part into small pieces. On the other hand, such parts as the African violet leaves can be used as a whole. Number 2. Using detergent and water, wash the plant part for about 20 minutes. Number 3. Transfer the plant part into sterilizing Clorox solution, shake for a minute and leave to soak for 20 minutes. Number 4. Using a lathe, gently discard the Clorox and retain the plant part in the container and then cap the container. And the final procedure involves transferring the plant material to a tissue culture medium. The process is 70% alcohol should be used for the sterilization of the equipment used in containers. First, open the container and pour sterile water to cover half the container. Second, cover with a sterile lid again and shake the container for 2 to 3 minutes in order to wash the tissue and remove the bleach. Third, pour the water and repeat this 3 times. Fourth, using sterilized gloves, remove the plant part from the container and onto a sterile petri dish. Fifth, using a sterile blade, cut the plant material to smaller pieces of about 2 to 3 mm across, avoiding the parts that have been damaged by bleach. Sixth and the final one, using sterile forceps, place a section of the plant into the medium. Let's talk about the technique for plant in vitro culture. Micropropagation. This technique is used for the purposes of developing high-quality clonal plants. This has the potential to provide rapid and large-scale propagation of new genotypes. Somatic cell genetics, used for haploid production and somatic hybridization. Transgenic plants, used for expression of mammalian genes or plant genes for various species. It has proved beneficial for the engineering of species that are resistant against viruses and insects. Now, the importance of tissue culture. Tissue culture is very important in biology due to its wide range of applications. Both plant and animal tissues can be used for culturing. For example, animal tissue culture helps in preserving an organ or tissue. Plant tissue culture may be used for genetic modification of a plant or simply increase its yield. The cells of the plants can be genetically altered to produce plants with desirable characteristics. This technique utilizes the plant's ability to rejuvenate the tissues rapidly. It produces exact copies of itself known as clones. It is a technique of quickly producing plants without any tubers, seeds or bulbs. It also helps in the conservation of plant biodiversity by the production of endangered plants. Let's learn about the one of main topics, that is its advantages and the disadvantages. Advantages of tissue culture. Number 1. The plantlets are obtained in a very short time with a small amount of plant tissue. Number 2. The new plants produced are disease-free. Number 3. The plants can be grown throughout the year, irrespective of the season. Number 4. A large space is not required to grow plants by tissue culture technique. Number 5. The production of new varieties in the marketplace speeds up. Number 6. This technique is being used for the production of ornamental plants such as dahlia, chrysanthemum, orchids, etc. Disadvantages of tissue culture is chromosomal abnormalities appear as culture age, that is undesirable changes, expensive, labor intensive especially if less than 1000 plants are needed. There are many applications of tissue culture, but let me inform you some major ones. Number 1. Used for the conservation of endangered plant species. Number 2. Large-scale growth of plant cells in liquid culture in bioreactors for production of valuable compounds, like plant-derived secondary metabolites and recombinant proteins used as biopharmaceuticals. Number 3. Used to cross distantly related species by protoplast fusion and regeneration of the novel hybrid. Number 4. Used for the production of identical sterile hybrid species. Number 5. Used for the large production of artificial seeds through somatic embryogenesis. Number 6. Used to produce plant resistant to the virus. And number 7. Used to produce improved nutritive value and vitamin quality of plants. Before moving further, subscribe the channel and press the bell icon to watch the latest video first. As I told, I will tell you interesting facts related to tissue culture. Let's go for it. Number 1. Plastic can contaminate cell cultures. Number 2. Frog nerve fibers were the first successfully cultured cells. Number 3. More than 32,000 papers rely on cell lines with no known original stock. Number 4. More than half of researchers fail to reproduce their own experiments. Number 5. The first immortal cell line HeLa was established without the donor's consent. Number 6. Almost 25% of all cell line contaminations are caused by HeLa cells. Number 7. Cell culture history. The first modern medium was created more than 60 years ago. Number 8. Components of culture medium can interact with each other and influence cultured cells. Number 9. Mini brains can be grown from pluripotent stem cells. Number 10. The year 2003 marks a milestone in bioprinting. Number 11. Israeli researchers succeed in creating a 3D printed human heart from fat tissue. Number 12. Engineers and cancer researchers team up to develop a new 3D scaffold. Number 13. Supercooling can increase preservation time of human organs. Number 14. Artificial intelligence just might interpret your next large data set in cell biology. Number 15. The 3D cell culture market could reach nearly $4 billion in 2021. I hope you have enjoyed the video. Tell me in the comment section on which topic you want the next video to be.